Good morning, students. So today we are with a new video of Clostridium perfringens. Now, before I move on to this particular topic, which is your Clostridium perfringens, I just want to say that Clostridium is having a huge clinical significance because some of the major pathogenic bacteria belongs to this Clostridium group. Like for example, if you heard about tetanus, right, the spastic paralysis which is caused by Clostridium tetani, or if I talk of your botulinum or botulism, that is the food poisoning or flaccid paralysis which is caused by Clostridium botulinum. So that's why Clostridium genera is also having a huge significance. Even if you talk about the commensal which is there in the gut, that is Clostridium difficile, so that also sometimes cause diseases when it finds opportunity. So most of the organism in Clostridium group are basically pathogenic microorganism, and they can even cause some devastating effect, even death. That's why for medical microbiology students, for clinical microbiology students, it is very essential to know about Clostridium genera. So today we will be discussing about Clostridium perfringens. This organism is a very important organism because it is associated with what is called as gas gangrene. So we often have heard about about sayings that when there is some kind of a deep wound or a deep burn, there comes the disease where you have necrotizing tissue. The tissues actually get killed, the tissues are degenerated by a bacteria, and sometimes you even have limb amputation because of this pathogen. So this organism is also called as flesh-eating bacteria, which is also called as Clostridium perfringens. Clostridium perfringens is also known as Clostridium welchi. Now moving on to its characteristic, in which one of the very important characteristics is that it is anaerobic, and this has led to more multiplication of this microorganism when you get to see them in the wounds. They basically hide in behind the clots in the wounds and they multiply, and because of the anaerobic environment, they are basically, their growth are basically favored. Apart from it, Clostridium over here is gram positive in nature. If you go with the Gram's character, you will get to see it is gram positive, and one of the major important feature of Clostridium is that it is spore forming. Spore forming means it can produce spores which help them to survive in the stress condition. Say it is dryness, say it is a presence of chemical, anything like that. And as we know that spores are actually located in three different parts. That is, sometimes a spore is centrally located. That means the spore is centrally located. A spore can be located in subterminal, that is, the in a cell, the spore, this endospore rather, is there in a subterminal position. So that is a subterminal pore. And even you get to see that the spore is at the termini. The spore is at the termini of a cell, and it looks like a badminton racket, which is your terminal spore. So in Clostridium perfringens we have two different kinds of spore, as we get to see, that is central spore. Sometimes it is seen that it is centrally located or subterminally located. Now for your information, I just want to highlight some few points. That is, when we talk of spore, you may be thinking that how can I distinguish spore or how can I visualize spore under microscope. So this can be done by the help of endospore staining, which is basically a differential staining which differentiates a vegetative cell from its spore by the color, as the vegetative cell takes the pink color or red color and the spore takes the green color. So from there you can get the location of the spore, whether it is centrally located or it is subterminally located or terminally located. Now comes the importance of other important factor, like for example capsulated. Again, students, when you have studied virology, your medical microbiology, one of the very important part is capsulation, capsule, formation of capsule around the microorganism. In my last video, I have talked about that this capsule can be a virulence factor because that hides the organism from the host immune system. So that's why presence of capsule is a very very important factor for pathogenicity, and here again in this pathogenic organism you have capsules. Moreover, they are pleomorphic. This is another important term which may be introduced. So this is actually pleomorphic, that is when the morphology is changed. Like for example, in amoeba you will not find any kind of morphology, they change their morphology, right? They have pseudopodia, they change their morphology according to their need. So that is basically called as pleomorphic, but they are non-motile, they are not motile, they do not have flagella. Now another important point of Clostridium that is already one should know, and it is like a awareness, right? Like these videos are not only a tutorial video for those who actually require the information, right? That like a video, okay, for a BSc student or a MSc student, right, who is studying medical microbiology, but simultaneously those who are MBBS student who actually are studying on various microorganisms in their microbiology, they can also refer to this particular video, right? Now I move on to another important point over here, that is what is the availability, means where this Clostridium basically thrives. This is a very very important point. Like Clostridium thrives mostly in the soil. Now we know that in most of the cases soil organism like Bacillus subtilis is under risk group 1, so it doesn't cause any kind of disease, but this soil is only the source of this Clostridium. That's why it says that even if you have a cut and your soil comes in contact with a cut wound, you may have to take tetanus toxoid. Otherwise, once it was said that even you have a cut with a rusted iron, then only you have to take tetanus toxoid, but now the concept is different. So it may be in the soil, it may be in the water they are present, even they are present in the gastrointestinal tract, sometimes causing food poisoning when they produce enterotoxins. Right? Now moving on to another important point, which is toxin, right? So toxins, which causes toxic effect. Toxins comes from the word toxicity, that is anything which is hazardous, right? So these toxins are basically produced by the microorganisms and they produce some of the other kind of hazards. So the toxins over here is classified into A, B, C, D, E. But I will tell you that generally in Clostridium perfringens the toxins are usually saccharolytic. Saccharolytic means they are degrading the carbohydrate, or proteolytic, that means they are degrading the protein. Now you tell me students that any kind of cell, they survive because of the presence of carbohydrate, one of the macromolecule, and protein is another macromolecule. So if your protein synthesis stop or your protein gets degraded, the cell will not be able to survive, and that results in necrosis of the tissue, necrosis of the cell, leading to the formation of gas gangrene, leading to the formation of more and more necrotizing tissue, where limb amputation becomes one of the important factor for survival of the patient, right? Moving on to the cultural characteristics. So this is the last part of this video, that is the cultural characteristic. Then in the next video I will be discussing the other points. So we usually use Robertson cooked meat broth for the cultural characteristics. Moreover, we get to see for the growth of the microorganisms, or apart from it, like they have, if you inoculate them in lactose, in litmus milk, it will have a stormy fermentation. So the lactose will be fermented to lactic acid, and this acid can coagulate casein and form casein acid clot. Apart from it, here I just give an example of certain test which is called as in-vic test, right? Please student refer in-vic test for enteric group of bacteria like Escherichia coli, Enterococcus and many other group of bacteria, where according to Burgess manual, this biochemical test is a very important test. That is in-vic, that is I for indole, M for methyl red, V for Voges Proskauer, C for citrate reduction. So here you get to see that this methyl red and Voges-Proskauer test is done for this sample or this organism, and your methyl red is positive for this particular organism and VP is negative. And even nitrate reduction test, which is another important test as per the Burgess manual, which is also positive in this particular case. So in a nutshell, in this video, if I point out the gist of this video, Clostridium perfringens is anaerobic, gram-positive, and it is spore forming. The spore may be centrally located, subterminally located. The Clostridium is present in soil, water, and sometimes gastrointestinal tract, and it is capsulated, which is one of the virulence factor, pleomorphic, having no morphology. It can change its morphology, and non-motile, that is, it doesn't have any flagella. It produces toxins and cause toxic effect, either saccharolytic or proteolytic, and it is further classified as type A, B, C, D, E. And if we move on to cultural characteristics, we can grow them in Robertson's cooked meat media, where there is a color change in the meat broth, and if you inoculate it into a litmus milk having lactose, it will have stormy fermentation, which will coagulate casein and coagulate this casein acid clot, and then it is, according to Invictus, it is methyl red positive and Voges-Proskauer negative and nitrate reduction positive. So with this, I am ending my video with 2 questions which can write down in your comment box. That is, what are the pathogenic microorganisms which are associated with Clostridium genera? Can you differentiate between spastic paralysis and flaccid paralysis? And the third question is, what is necrotizing tissue damage? So these are the 3 questions, my viewers. If you can answer this question, please write down in the comment box. Thank you so much.